Thanks, Greg, and thanks for the preview. I'll be touching on a few of these things as I go along. This is what I'm going to present today. Well, first is the iron deficiency chlorosis, which is of interest in the eastern part of the state. And then just basic nitrogen, phosphate, potassium, and then finally some sulfur micros other than iron. So that'll fill up the 20 minutes, I think, really nicely. So if you're in eastern North Dakota, this is a big deal. That the chlorosis is fence row to fence row. It can be pretty devastating if you have the wrong variety and all the stars are not aligned correctly. You'll struggle to get more than 10 bushels an acre, I think, if you have it really severely. So you see the chlorosis on the younger leaves. You notice that in this picture, and I think this one actually came from the Carrington area a number of years ago during a wet season. The unifoliate leaves, the monofoliate leaves, they remain green. Cotyledons are green, monofoliates are green. So until this stage, the iron that's in the seed actually goes up to the leaves. But as soon as the first trifoliates come out, the physiology of the soybean changes. And from then on, the trifoliates that come out are very susceptible because iron is no longer mobile within the plant and it has to continually take it from the soil. The root systems are very small at this stage, and if conditions are right, you have the wrong variety. If you have carbonates and all those things, then you can get this kind of thing. Notice this interveinal chlorosis because the veins tend to remain green. If you center in on that center plant, you'll see the veins are still green, and between the veins, the main veins, the leaf is yellow. So that's interveinal chlorosis, IDC. So the only cause for this is the carbonates. It's the only cause. Other things factor in the severity, but the only cause is carbonates. So if you have pHs below 7, there's no way that you're going to get IDC. If you have pHs above 7, that means that you have carbonates in your soil. People that zone sample, and that's really a lot of people within the state, they understand that you have areas in the field that are fairly acid and areas in the field that have maybe pHs, as Greg was talking about, that approach 8 or maybe even above, really in the same field. So zone sampling is really important to get a handle on what the status of pH is in the field. If you have areas in the field that have pH, regional soil testing labs can analyze those to determine the percent carbonate. It's not a standard test, but certain labs can do that, and it's not that expensive to try to figure out what you have. So the soybean, the strategy that soybeans use in order to get iron is to acidify its root zone, and it doesn't do that. Yeah, it influences solubility of iron compounds a little bit, but the big reason why it does it is that the iron in the soil is what we call oxidized iron. It's exposed to air, and so it's a form of iron that the solubility is very, very, very low. And what the soybean root does when it acidifies its root zone is it sets the stage for a protein it exudes in order to change the oxidized iron to reduced iron and increases the solubility about 1 trillion times. So it's really, really important. What the carbonates do in the soil, when they dissolve, they form bicarbonate, which is that HCO3 minus down on the bottom there. And that's what neutralizes the acidity around the roots. And the more carbonate and bicarbonate there is, the harder time the soybean has to make sure that protein works. And at some point, it just gets overwhelmed with too much carbonate or other stresses in the plant. So if the carbonates are present and the environment supports IDC, and what's the environment? That means if the soil is moist, a lot of times if you get a dry spring and you don't really look like you're going to have a lot of IDC, and then you get a little bit of a rain and all of a sudden the field turns yellow overnight, or parts of it does, that's the moisture. It's dissolving the carbonate into bicarbonate, and it's neutralizing so wet soils, cooler soil temperatures, and even herbicide stress. Now that we're in the post Roundup era, the herbicides we use are a lot more stressful on the plants than the Roundup used to be, and so you actually get that too. The huge stress, one of the huge stresses on the soybean plant is the salts. The soybeans originate from an area in eastern China, not along the coast where there is salts, but interior where salts are really kind of non-existent. And so it doesn't really have this innate ability to overcome salts. This is some work by Kirsten Butcher, is now a a professor in our department, and this is when she was a master's student. She did some work on IDC and soybean yield. And if you get down to maybe a EC of 1.5, you've already clipped about 15% off your yields. Not even with IDC. I mean, IDC isn't present in this study, and still, just the salts alone clip the yield by 15%. And then you add IDC susceptibility with the salt, and you got a major issue. So these are strategies that I would strongly recommend for IDC reduction. One is field selection. Not all fields are fit to grow soybeans on. If you have a field that has, say, ECs of 2 and above, I can't imagine any scenario where you're going to get a profitable soybean yield off of that. So you need to do some serious screening. You can't just throw soybeans anywhere. The other is the variety selection. That's really, really important after the field selection. And as Greg showed, we have really good screening in this state for IDC that's relevant to this state. Other states have screenings, but they don't have the salt stress that we do. And so you can't use southern Minnesota or Iowa or Illinois or Wisconsin IDC measurements in order to try to figure out what your tolerance is. You have to use the state measurements where the salts and the carbonates are together in our conditions. So use our variety selection from NDSU. It's a tremendous resource. Next, you can put iron fertilizer on, but you just can't throw anything on. The iron fertilizers that work the best, the main one and the one that's tried and true and is the ortho isomer of EDDHA chelate. And the other one that's not quite as well researched, but Jay Goose, if you remember Jay, he did quite a bit of work on fertilizers for IDC, and the other one was iron HBED. And I'm not sure how prevalent that is in the market, but he had good results with that also at the end of his career. The other is companion crop, and I'll show you that in a minute. And the other is maybe wider rows, but with the weed spectrum we have, that's probably a doomed recommendation. So that's only if you don't have any weeds. And I don't know anybody that doesn't have any weeds. So let's move on. So field selection, you want to pick fields with low carbonate levels or slightly acid soils. That's preferred, pH less than 7.2. Try to select fields with EC no higher than 1.5, and then select fields with decent drainage. And that all three of those things will help quite a bit. This is the difference between ortho on the left and ortho-para on the right. Whenever you manufacture an organic compound, you get these isomers coming out, and there's no way around it. Even the best iron ortho EDDHA has a small component of ortho-para. You just can't get around it. It's just part of it. So the ortho-para can kind of flash a little greenness to the plant really quickly, but then it's done. It doesn't have the ability to take iron from the soil solution and have it available to the plant anymore. Whereas the ortho, because it has 1, 2, 3, 4, 5, 6 points that hold the iron, it has enough power that once it dumps the iron off at the plant root, it can go back in the soil solution, pick up some more iron, and so its use is a lot better. Its performance is a lot better. So the pot experiment on the bottom, which is something that Jay Goose and Sarah Lovis, when she was a graduate student of Jay Goose, they did. This product is only 1.5% ortho with these specific additions to the plots, and with the same additions to the plot with a 5.5% ortho, which would be the material that would be in soybean, for example. You can see the difference between the ability to overcome IDC on the left compared to that on the right. All right, so companion crop, why does this work? Another factor that influences the IDC is high soil nitrate values. So once you get above maybe 80 pounds of N in the top, a couple feet or so, you have more IDC in that situation as long as you have the carbonates and the moisture around to make it so. It doesn't have anything to do with the iron uptake, but it has a lot to do with what happens physiologically within the plant. The other thing is when you put a companion crop on, as I think you would appreciate, the oats in this case are taking up water too and drying the soil off compared to what it would be if there wasn't any companion crop. So it's taking up nitrate, lowering the risk with that, and it's taking up water and lowering the risk for that. And some of the results have been done in a wet year are pretty astounding. There's some work from Minnesota. North Dakota and South Dakota were also involved in the study, but we didn't have quite the wet year that they did in Minnesota, and we didn't have quite the results. But when the spring is wet, it really, really helps. The next thing I'm going to cover is using an inoculant. If you have a field that's never seen soybeans before, you just have to do it, and you double inoculate. And I don't mean using twice the rate. I mean using two different modes of action. Maybe using a peat on the seed or a liquid with the seed, and then putting the granular down as within the furrow. That's what I mean by double inoculation. So it can really really be helpful. Notice that both of them in this Minot study is numerically. Greg's going to give me a hard time for that because it's not statistically different. But the biggest thing is that it's easy to mess up the first time when you're using an inoculant, but when you're using two different types of inoculants, the ability to mess it up is greatly reduced, and that's really the biggest thing. You know, if you really try hard, you can mess it up, but I don't think any of you would do that. So once you inoculate, when do you have to inoculate again? This is some work that's in the Soybeans Fertility Bulletin circular that we've had out for several years. And after 1 year, you can see that there's a lot of bradyrhizobium cells, little bacteria, per gram of soil. That's per gram of soil. You can imagine how much is in an acre. But after 2 years, it's still pretty substantial, isn't it? So our general recommendation is that if you had successful inoculation the first year, that if you grow soybeans within 4 years, that you really shouldn't need to. After that, you can, but it's not that expensive, but you would still be fine if you didn't. The next thing is nitrogen for soybean. There's no need to put on nitrogen for soybeans. So the most recent work was with Chris Augustine and his PhD work in the northwest part of North Dakota. We didn't see a whiff of any help, whether we fertilize with 50 pounds of N or not. It's just a drain on expenses, so just don't do it. No need. Also, a group of us got together at Carrington half a dozen years ago, including Chris's work up in the northwest part of the state. But researchers at Carrington and other places around the state have done phosphate work with soybeans, and we came up with a new recommendation that we only needed phosphate if your soil test was 7 ppm or less. Broadcast or band seed separated, as Greg was pointing out, not with the seed, but separated. Seed place P doesn't increase yield in most trials, and it can decrease yields, especially if the soil becomes dry after seeding. So we definitely don't want to recommend that. So right now, these are our phosphate rates, and we kind of sold those for the old recommendations that we had before because we didn't have any new data. So in very low testing soils, 0 to 3, I've never seen a 0, but from 2 to 3 ppm, about 100 pounds of MAP per acre, and then 50 pounds if you have a little bit higher than that. And if you have 8 or above, you don't need any at all. So Lindsay Malone, who's a new hire, this will be her second spring. She's working with several of the RECs, and she shared this data with me yesterday. These are her sites, one down in Fairmount, southeast part of the state, and then A high P, that means it has P greater than 7 ppm, and then a low P, which is less than 7 ppm in the Barney area, which is what, just west southwest of Morton. And so we're trying to figure out, she's trying to figure out, and her colleagues are figuring out what, if those rates are appropriate, that 100 pounds of MAP and 50 pounds of MAP, are they really appropriate, or do we need to go to something else? So this is just preliminary. My guess is that when it's all said and done, it'll be related to relative yield. You can see the Fairmount site and the high P site, that the response curve is pretty close to the same, even though the yields are different. And then the site in the on the right hand side, not much difference there at all, but the yields are really, really low. Kilograms per hectare is about pounds per acre, if you want to just kind of more or less. So what, 1,000 pounds is what? It's what, 15 bushels, something like that. So that's pretty pitiful yields. You know, particularly dry year or something like that. So phosphorus really didn't mean anything with those, but the other two sites it did, and it didn't seem to be related to actual yield. It was related to relative yield. All right, so that's that. This is some work by Greg and Paul Hendrickson, where they looked at the stand of soybeans when you put fertilizer in the furrow, and when you had a 2x2 band in that second row, it didn't affect the yield plus or minus at all. But when you started putting it in the furrow, you reduced stand a lot and you reduce yield a lot. And so the reason we put on fertilizer is to increase yield, not decrease it. So don't put fertilizer in the furrow unless you're dealing with iron. Our potassium rates are intercircular as well. I won't go into great detail with those. The number on the left on the bottom there is with the lower smectite to illite ratio clays, and the one on the right is with the higher smectite to ratio clays. We have a circular that has the MAP in it. This is the MAP. The dark areas are the higher smectite to illite. That makes a difference because the smectites can temporarily retain or fix potassium, and the illite clays, they just shoot potash out whether it's wet or dry. So they're more forgiving and the soil test level can be lower. That's why the two recommendations. As Greg said, the in-season foliar sprays almost never effective, paying back what you spent if doing anything. So the work that Chris Augustine did in northwest part of North Dakota certainly showed that, and a national study that Hans was involved in also showed that. In that publication, we had with Chris's work, he had 2 treatments at each of his sites in north central and the northwest North Dakota, 6 site years, no effect on yield at 2 different timings from 2016 to 2018. It's not necessary. Soybean is much more able to extract sulfur from the soil compared to wheat or corn. Once in a while, you find someplace, the most recent one in Indiana, that I read about in either Agronomy Journal or Soil Science Society. But normally the soybeans, even if you have sulfur deficient corn or small grains or something right next to it, the soybean is perfectly happy. How that happens, I have no idea, but it does happen. Also, I'm not sure that I would really put any sulfur on the soybeans at all. Because it's possible to rescue the sulfur deficient soybean. If you see that the upper leaves are turning yellow and it's not the interveinal IDC stuff, you can certainly rescue it because soybean's an August crop. It doesn't have to be continually nurtured like corn or small grains do. And we don't find any need for micronutrient supplement in North Dakota except for iron. So here's the summary. No nitrogen required. Inoculation only when years are separated widely or the first year. The critical value for Olsen P is 7. No fertilizer with the seed except for perhaps an iron supplement. The potassium is related to clay chemistry. Manage IDC with a multi-prong approach. Monitor soil pH so it doesn't drop below 5.7. Sample by zone to figure out if that's happening or not. And sulfur is probably not an issue for most of you. There's my contact information, and I think I'm done, Greg.